Okay, hello again. As I promised last time, we have one more group of molecules or compounds to talk about their metabolism, and today we're going to talk about them, the nucleotides. And once again, I like to show this figure and involve or many of the metabolic pathways in any given cell. Um, these are mostly what we sometimes call housekeeping pathways, so things that all cells need in order to function. So it does not include any kind of specialized activity that would be in any specific cell type. But again, the shaded area here shows you the reactions uh, for the metabolism of nucleotides, and they're right in the middle of the uh, reactions containing nitrogen, or molecules containing nitrogen. But again, you can see how they fit into other pathways, including glycolysis and eventually into the TCA cycle if we want to go that far. So when we talk about the synthesis of nucleotides, there are two pathways. The de novo pathway, which uses intermediate compounds, mostly ones you should be familiar with by now, to synthesize new bases and new nucleotides. But there's an alternative pathway, the salvage pathway, which is involved in recycling nucleotides. And although we can make as much or as many nucleotides as we need, if we can't carry out these salvage pathways, it can lead to some serious medical issues. So there are two types of nucleotides, two-ring purines shown here, and the one-ring pyrimidines that we'll talk about in a couple minutes. But these are four examples of purines. On the right are intermediates in purine synthesis, hypoxanthine at the top and xanthine at the bottom. The difference is the extra oxygen, which is at the number two carbon. Then if we replace the oxygen on hypoxanthine with a amino group, we get adenine. If we replace the alternative oxygen on xanthine with an amino group, we get guanine. And so these all just differ in their side moieties. And even though they're very similar molecules, they do have different properties. Again, adenine and guanine are found in both RNA and DNA, whereas the other two molecules are intermediates in their synthesis and, as we'll see, in degradation. Okay, this is where all the different atoms of the two-ring structure come from. You see the green area is the glycine backbone between the two rings. There are a couple of methyl or single carbons donated by the active form of folic acid, tetrahydrofolate. A couple nitrogens that derive from glutamine, and another one from aspartate, and then one last carbon that is donated, is from directly from carbon dioxide. So this is the pathway for synthesis, and again, we're not going to belabor this. We don't need to sit down and memorize it, but I think just go through it quickly so you get an idea of what's going on here. So we start up at the left-hand corner with the ribose that's got three phosphates attached to it, and so it's phosphoribosyl pyrophosphate, or PRPP. So it has two phosphates on what's called the 1' carbon and one phosphate at the other end of the molecule at the 5' carbon. Okay, so the base is going to be attached at at the 1' carbon where the two phosphates are. And so the first step is to transfer the amino group from glutamine to that carbon on the ribose, eliminating the pyrophosphate, the two phosphates, and producing glutamate. So this is obviously a step that takes some energy. The next step also requires ATP, and now glycine is added to the amino group that was added in the previous step. At that point, we add a methyl group, again donated by tetrahydrofolate, and then we can complete the five-membered ring, close the five-membered ring, at the same time adding another nitrogen to start the six-membered ring, or the other ring. Then CO2 is added, and nitrogen is added to that, donated from aspartate, and when it then gets cleaved off or deaminated, producing fumarate, which, as we all remember, is an intermediate in the TCA cycle. And so we'll come back to that. Add another methyl group, and we're ready from tetrahydrofolate, and we're ready to close that ring as well. So now we've ended up with inosine monophosphate. And if you look closely and you compare it to the previous slide, I guess 2 slides ago, you'll see that that base looks like hypoxanthine. So when hypoxanthine is by itself, just the base, that's we call hypoxanthine. When you add a sugar and a phosphate, now it becomes inosine monophosphate. So inosine when there's a ribose attached to it, hypoxanthine without the ribose. A little bit of nomenclature to confuse us all. Okay, this is in a little more detail. I'm not going to go through this. I just want to point out the first step here, and that is to take the ribose 5-phosphate, so with a phosphate on the 5' end, and use ATP to add the 2 phosphates at the 1' carbon, and that will be the site. Then gets amino group added, cleaving off the pyrophosphate or 2 phosphates in the process. Now once inosine or IMP is formed, it can split into 2 different pathways, and that's shown here on the left. An amino group again is exchanged for the oxygen to produce AMP. On the right, first we have to add the extra oxygen to make xanthine monophosphate, and then that is exchanged for an amino group to make GMP. And so then we have our precursors for RNA or DNA. Now, like other pathways, this is highly regulated, and there's a, as you might imagine, as in most pathways, the regulation usually makes some sense, just like it does here. At the right side of this figure, we see that both AMP and GMP are inhibiting the production of themselves, so that it can maintain the appropriate ratio of AMP to GMP. So if we have more AMP, that will shut down the synthesis of itself, so that we make more GMP. If we have an excess of GMP, then we'll make mostly AMP. There's also regulation upstream. At the very start, both nucleotides feed back and inhibit the production of PRPP. So if we have an excess of nucleotides, it will shut it off at the first step. Now, if we do get a lot of PRPP, that's a positive regulator to send it through to make the nucleotides. But also an excess of G or A will inhibit that as well. So regulation at several steps here, both in the production of the precursor inosine monophosphate and also to control the ratio of G to A. So that was the de novo pathway, and we also have a salvage pathway, and that's shown here for both guanine and hypoxanthine, and it's shown together because they both use the same enzyme. So basically, if you have either one of those bases, you can add it to PRPP to make a IMP or GMP, and that uses the enzyme hypoxanthine guanine phosphoribosyltransferase transferase. So these enzymes called phosphoribosyltransferases transferases put a base on the sugar phosphate, but it has to have the two phosphates at the one position in order to do that. And we'll come back to this because if you don't have this enzyme, you're going to have some very severe medical issues. So even again, we can make as many nucleotides as we want. Without this enzyme, things don't work quite right, as we'll see. Now, if we have an excess of nucleotides, we don't know what to do with them, we need to get rid of them, and these are the pathways to remove nucleotides from our system. If you eat a lot of rich foods, some foods are very high in nucleotides, so you might need to get rid of some of these. And let's look at the one at the bottom first, but they're all heading to the same final product, and that's uric acid, shown on the right. And if you look at it closely, you'll see that an extra oxygen has been added to the 5-member ring here, making it more soluble or more hydrophilic, and that makes it more readily excreted through the kidneys. And that's the whole idea, is to make this compound easier to go through the kidneys and get excreted. So at the bottom, basically the first step is to remove the phosphate from the sugar, then remove the sugar, leaving the base, and then guanine is converted back to xanthine. As you remember, xanthine was the final precursor for guanine, and then that gets oxidized to uric acid. Now, if we have xanthine monophosphate, the same pathway, remove the phosphate, remove the sugar, and then we oxidize the xanthine to uric acid. Now, AMP is going to ultimately go through the same process, but there are two pathways for breaking down or converting AMP into uric acid. One that deaminates it first to make IMP, and then gets rid of the phosphate, then the sugar to make hypoxanthine, and then it has to be oxidized twice to get the uric acid. The other pathway takes the phosphate off first to make adenosine, that's the base plus the sugar, and then deaminates it. And this is and so we get the inosine, just like the first pathway. And once again, if we don't have that enzyme, adenosine deaminase, there are some severe medical issues that we'll talk about in a minute. So even though you can get rid of AMP through AMP deaminase, if you don't have adenosine deaminase, there's some severe medical consequences. This is the mechanism of xanthine oxidase that oxidizes xanthine into uric acid, and I don't really want to belabor this. It's a somewhat unique enzyme that requires a molybdenum to transfer the oxygen to the xanthine. And if you'll notice, the structure looks a little bit different than the one I showed you on the previous slide. We've got a hydroxyl group added and not a carbonyl or double bond oxygen. But that hydrogen quickly gets transferred to the nitrogen, and the double bond goes between the oxygen and the carbon. So these are tautomeric forms, but the double bond oxygen is much more stable. And just like we talked about way back when, oxygen just really loves electrons and really good at sucking them up. So it's gonna pull those electrons towards it and make the double bond there. Now, if we have an excess of uric acid, that creates some problems, and mostly in the joints, and because it gets deposited there, and it can cause inflammation, which is basically called gout. And gout can be due to difficulty in excreting uric acid or in excess production of uric acid. So in both ways you get an accumulation, and probably in some cases you get the accumulation due to both of these issues. So it then accumulates as a sodium salt, sodium urate, in the joints, and where it's phagocytized by neutrophils. And these crystals, crystalline urate, damage the neutrophils, releasing contents and causing this inflammatory response, which is very painful. I've been told that people with gout, and the first place it usually shows up is in the big toe, the picture I just showed you, and that it's so painful that people can't even have a sheet over their foot at night. Now, there are some drugs, at least one drug that's used to treat this, and this is the drug allopurinol. You can see it's very similar to hypoxanthine, except that the nitrogen is has been switched with the carbon, where the oxygen is usually put on to make uric acid. So this compound inhibits xanthine oxidase, which is the final enzyme in the production of uric acid. And this shows again xanthine oxidase actually oxidizes the compound in two different places. So it has two different activities to put oxygen on hypoxanthine in both extra positions to make the uric acid. So this shows the degradation of purines and the basis for allopurinol treatment. And again, you can see all these compounds, the degradation leads to xanthine, and again, allopurinol inhibits both the reactions that xanthine oxidase carries out. Now we're talking about HPRT, and that's, again, it's enzyme in the salvage pathway for both guanine and xanthine, so or hypoxanthine. And this is a diagram showing the synthesis and then breakdown of guanine. So starting with ribose 5-phosphate and going to uric acid. And so in the middle here we have the nucleotide. And this shows if you have hypoxanthine or guanine, the HGPRT enzyme can ribosylate that and form back the nucleotide. If you don't have that enzyme, the HGPRT, you can cause some very serious issues, which is called Lesch-Nyhan syndrome. And the symptoms are crippling gouty arthritis, so it's a really terrible form of gout, basically, which you might imagine it would have similar effects that gout has. But it also has severe malfunctions of the nervous system, leading to mental retardation, spasticity, sometimes aggressive behavior and self-mutilation. And the self-mutilation really comes from the fact that these people can't stop eating, and if you take the food away from them, they just start chewing on their lips. Now, HPRT gene is located on the X chromosome, so that should tell you right away that we only find this disease in males. Now, if it were a genetic disease on the X chromosome that didn't manifest itself till later in life, we might find females that at a lower rate than males that would have the problem. But these people don't live that long, they don't reproduce, and so since males get their X chromosomes from their mothers, and the female gets one X chromosome from the father, the father will never donate a Lesch-Nyhan chromosome to a daughter. Again, this is a recessive problem, so you need two bad copies of the HGPRT gene in order to have Lesch-Nyhan syndrome. Now, mention that the lack of adenosine deaminase causes problems, even though there are other pathways to get rid of AMP. But people with adenosine deaminase deficiency or ADA deficiency have what's called severe combined immunodeficiency syndrome or disease. And that's as opposed to acquired immunodeficiency disease or AIDS, which is caused by a virus. This is a genetic disease that basically results in the same problem. If you don't have adenosine deaminase, it affects the thymus. Basically, you don't have any thymus because the thymus is basically just a sack of cells that are maturing into T cells, which are important in the immune system. So without ADA, you don't get any T cell production and therefore have no immune system. Now, those people, if they catch it early enough, can survive in isolation, but could never be allowed to go out into the world where they would quickly contract something that would succumb them because they have no means of no defense, no immune defense against it. Okay, I want to touch on this again. I mentioned it earlier, but basically in the production of purines. We do convert aspartate to fumarate when it donates the amino group, and that fumarate, again, as we all remember, is an intermediate in the TCA cycle or citric acid cycle, and so in that way we can feed molecules into the TCA cycle through the production of nucleotides. Okay, so just to mention here that different animals do excrete these compounds in different forms. Humans, like primates, as well as some other animals, excrete it as uric acid, but there are a number of breakdown products of that other animals use to get rid of the nitrogen, including some animals that break it down all the way to ammonia and excrete it as ammonia. Okay, I'm going to move on now to talk about the other group of these are the pyrimidines. These are a single six-member ring, and here are 4 examples. Start with the lower right-hand corner. We see the precursor orotic acid, and that then can be used to make the 2 bases found in RNA, uracil and cytosine, as well as the bases made in DNA, which is, again, is cytosine and, of course, thymine. If you notice the structures here, the difference between uracil and thymine is a methyl group attached to the ring, and so that's the only difference. The only difference in the bases between RNA and DNA. Okay, so when we were talking about the urea cycle, I mentioned that we would see another enzyme that had the same activity and the same name as the one of the enzymes in the urea cycle, and this is involved in the first step in pyrimidine synthesis. This is the enzyme carbamyl phosphate synthetase. And so it makes the molecule carbamyl phosphate from carbon dioxide, amino group from glutamine, and it requires 2 ATPs. One of the phosphates gets attached to the molecule, and this is the mechanism. So we'll see in a minute here why we need 2 different enzymes to carry out the same function, one for pyrimidine synthesis and one for the breakdown of nitrogen into urea. So these are, this is the pathway again for making pyrimidines. Again, the first step is carbamyl phosphate synthetase to make carbamyl phosphate. Then we add aspartate through aspartate transcarbamylase, and finally we can then complete the ring using the enzyme dihydroorotase. And then a couple of modifications to get to orotic acid, which then is added to the sugar phosphate. So notice this is a little different than purine synthesis, where the base was built onto the sugar. In this case, the ring is made first and then added to the ribose. Once we have that, then we can convert those to the other nucleotides. Now, it turns out the first 3 enzyme activities in this pathway, the carbonyl phosphate synthetase, aspartate transcarbamylase, and dihydrooroutase are all found on the same polypeptide. That was actually determined because there's a inhibitor of the second enzyme, the aspartate transcarbamylase, that was being looked at as a possible treatment for cancer. You know, if you can't make pyrimidines, you can't make DNA, and so that might inhibit cell division. And after it was learned that you could select out cells that had extra copies of the gene by treating them, by growing them in the presence of the inhibitor, that was done with this inhibitor called PALA. And so they amplified up the number of genes for aspartate transcarbamylase. And when they did that, they found that they also got extra genes for carbonyl phosphate synthetase and dihydrooroutase. And so then they realized that it was the same gene making one polypeptide that had all three of these activities. So now you can see why you wouldn't want to use the same enzyme in the urea cycle, because it's attached to these other two enzyme activities in the pyrimidine synthesis pathway. Turns out the last two steps in the pathway are also on the same polypeptide. Together on another polypeptide. So unlike purines, which are made up of from a number of different molecules, the pyrimidines are much simpler, where we just take a molecule of aspartate, add in a carbon and nitrogen from carbonyl phosphate, and make the six-membered ring. Once we have that, they can be interconverted, so you can convert one to the other. And all these reactions that involve nucleotides and modifying them from one to the other, or whatever, occur at different levels of phosphorylation. So this example, it's in a triphosphate form, but some are at monophosphates and some are diphosphates. And we're not really going to memorize those for sure. It's just interesting that in some cases you have one step acting at the triphosphate level, and the next step may act at the diphosphate level, so you have to drop off a phosphate in order to go on to the next step. Now, a conversion of the deoxy versions of these happens at the monophosphate level. So again, the point here is that if we have one nucleotide, we could potentially convert it to the other ones. And you saw that with the with the purines, because all the nucleotides could be converted back into hypoxanthine or xanthine, and then again, that's a precursor for making them. So once if you need to make some more of one nucleotide, you can do it at the expense of the other ones. Now, one other modification we need to make to the pyrimidines is to methylate DUMP to make TMP, and that's the pathway shown in this figure. It looks a little complicated, but it's maybe not quite so bad. So up at the top is the enzyme that does that, thymidylate synthase. Simply adds the methyl group to the UMP to make DUMP to make DTMP, which we usually just call TMP because you don't ever find the ribose form of that. And the molecule that donates the carbon or the methyl group is the active form of folic acid, tetrahydrofolate, shown beneath the UMP here. And tetrahydrofolate is the single carbon donor for many reactions in the cell. What's unique about this reaction is, unlike every other reaction that involves folic acid or its active form, tetrahydrofolate, the tetrahydrofolate is oxidized back to dihydrofolate. Like I said, in all other reactions that involve tetrahydrofolate, that does not happen. Okay, so it has to be re-reduced before it can be used again, and that uses the enzyme dihydrofolate reductase, which actually converts folic acid to tetrahydrofolate as well. So dihydrofolate reductase has been a target for therapy because if you can't re-reduce the tetrahydrofolate, then you can't make thymidine, and so then you can't make DNA, and so you can't grow. And actually, if you treat cells with inhibitor, which we'll see in a minute, of dihydrofolate reductase, or DHFR, what happens is the cells continue to grow. In other words, they tend to keep getting bigger and bigger, but they can't divide because they can't make DNA, and so they eventually get too big and they just pop. Anyway, there are a number of molecules that inhibit dihydrofolate reductase. 3 shown here, 2 at the top that vary just at this position R, where the R group is. If there's a hydrogen there, it's aminopterin. If there's a methyl group, it's amethopterin, or more commonly called methotrexate. And these drugs, particularly methotrexate, has been used to treat cancer for decades. And this would be an example of a nonspecific approach or therapy because it just inhibits the growth of cells. So the idea is that the cancer cells are growing faster than normal cells in the body, and so they will be more greatly affected by the drug. There's another inhibitor that has part of the folic acid, and that's shown in the bottom, trimethoprim. And this molecule inhibits the bacterial dihydrofolate reductase, but not the human or mammalian enzyme. And so it's been used as an antibiotic. It was also used to isolate the dihydrofolate reductase gene, which was one of the first genes ever isolated back several decades ago. At the time, the only other genes that were isolated were those that were produced in very large amounts in specialized tissues like the globin in red blood cells. And this was the first housekeeping gene, or gene that was expressed at very low levels in different cells to be isolated. And the way that was done was to take cells, in this case mouse cells, that made a lot of dihydrofolate reductase RNA, convert that into DNA, cDNA, or complementary DNA, and add it to E. coli, and then select for those cells that could grow in the presence of trimethoprim. So, and that was done, and many cells were isolated, grown up, and they contained a copy of the mouse dihydrofolate reductase gene. Now, there are also analogs of tetrahydrofolate that inhibit the synthesis of folic acid in bacteria. These are the sulfonamides, which have also been used as antibiotics. In fact, they were the first antibiotics that were available. And nowadays, they're still used and often in conjunction with trimethoprim. So you have two antibiotics that inhibit different steps in the same pathway. The sulfonamides inhibit the production of folic acid, and trimethoprim inhibits the reduction of folic acid to its active form, tetrahydrofolate. We also have analogs of thymidine that inhibit thymidylate synthase and have been used to treat cancer. And here's some examples. The one that's most commonly used is a 5-fluorouracil. And if you look closely, you'll see that there's a fluorine shown here, the orange circle, where the methyl group generally is in thymidine. So what happens is if you treat cells with 5-fluorouracil, the folic acid will try to transfer and the enzyme thymidylate synthase will try to transfer the methyl group to the pyrimidine ring. It'll attach it, but you need a hydrogen in order to release the rest of the folic acid. And with a fluorine there, there's no hydrogen to carry that out, so it stays attached and therefore destroys the pyrimidine as well as the folic acid. Now, degradation of pyrimidines is fairly straightforward. Both cytosine and uracil are converted into alanine plus some ammonia and carbon dioxide. Thymidine is a little trickier because it has that extra carbon, so it is converted into amino isobutyric acid, which is just alanine with a methyl group on it. Now, there's one last process we need to look at, and that's the conversion of the ribonucleotides into deoxyribonucleotides. And so you can see in this figure the reaction which reduces the hydroxyl group at the 2' position, shown in the, I guess that's kind of a reddish box, to a hydrogen to make the deoxyribonucleotide. And the same enzyme does this for all 4 nucleotides, and that's the enzyme ribonucleotide reductase. So this is the reaction, again, converting the hydroxyl group, reducing it to a hydrogen at the 2' location on the base, on the sugar. It oxidizes a thioredoxin, which then needs to be reduced using NADPH. So this is a schematic of ribonucleotide reductase, and it's a tetramer consisting of 2 alpha subunits and 2 beta subunits. The active site of the enzyme is between the two, shown here with this little red part of the tyrosine. And this is a highly regulated enzyme, and as you can imagine, so it's regulated in 2 ways, basically. One is by measuring the amount of deoxynucleotides available, and so if there's a lot of them, it will shut this down. If there aren't enough, it will activate it. And also by measuring the ratio of the nucleotides. So if there are a lot of G's, it will make A's. If there are a lot of C's, it will make T's, and so on. So at the bottom of the slide is a list of regulatory elements, and you can see there are 2 sites on the alpha subunit, one that that determines the amount of activity, the A site, and then the S site is determining which nucleotides to focus on. Now, if you break open the cells and take out all the nucleotides, what you find is they're pretty much all ribonucleotides, very few deoxyribonucleotides. And in fact, I was doing a project one time where I was measuring the amount of ribonucleotides, and we just ignored the deoxys because they were insignificant. And so what happens is they're pretty much made as needed, and the enzymes involved in that are associated with the replicating complex. So they're just form a giant complex that makes these nucleotides as the polymerase moves along the chain, synthesizing the DNA, and gives them to the polymerase as needed. So this figure is a schematic of the interconversion of these molecules, these purines. And remember, we have a number of inhibitors that inhibit dihydrofolate reductase as well as thymidylate synthetase, and so those will prevent DNA replication, and therefore cells can't divide. Remember, many of these inhibitors have been used to treat cancer, and the inhibitor of dihydrofolate reductase, trimethoprim, has been used as an antibiotic since it doesn't affect the human enzyme, but just the bacterial enzymes. Now the next set of equations just shows that we can pass these phosphates around between the different nucleotides, and so we can, if we don't have enough of one and we have plenty of the other, we can move the phosphates around. I want to just briefly mention one other thing, and that is to look at an analog of purines, and that's the psychoactive stimulant caffeine. You can see here it has the two-ring structure that purines have. Did I say pyrimidines last time? It's a purine. It also contains some methyl groups that have been added. And this is a drug that's found in a large variety of plants, particularly at high concentrations in coffee, as we're all well aware of. But it appears to be a natural pesticide that the plant uses as a defense against insects. But the vast majority of people in this country consume caffeine or other similar drugs every day. And again, you can see the similarity between caffeine and adenine. And this is an antagonist of adenosine receptors. And adenosine is a very versatile molecule. Of course, it's incorporated into DNA as well as RNA, and it's used to transfer energy or used to provide energy in many, many enzymatic reactions. It's also used as a messenger in the form of cyclic A, and it's also a neuroreceptor. And so caffeine is an antagonist for adenosine receptors. Also, it inhibits the enzyme phosphodiesterase, which causes the breakdown of cyclic A. So it will increase the levels of cyclic A in that cell so that it provides a much stronger signal. Okay, so next time we're going to move on and start talking about DNA replication.